Hello everyone, my name is Alnaj Thange from Shivajirao S. Jondle College of Pharmacy, Assangaon, studying in final year B Pharmacy. Today I am here to give some information about the Benin Casa hispida, the therapeutic power of ashgard, a comprehensive review on its pharmacological and health promoting effects. Here are the some key points which we are going to learn. Abstract. Ashgard, Benincasa hispida, also known as winter melon, it is a versatile plant valued for its nutritional and medicinal properties in a traditional medicine system. It is rich in vitamins, minerals, and bioactive compounds such as flavonoids, alkaloids, triterpenes. Ashgard also exhibit a wide range of pharmacological activities. This review article aims to explore the therapeutic potential of ashgard, focusing on its antioxidant, anti-inflammatory, anti-diabetic, and anticonvulsant, antihistaminic, anti-ulcer, antibacterial, antifungal, anti-asthmatic, diuretic, protective effect on Alzheimer's diseases, anti-aging properties. By highlighting recent scientific studies and traditional application, these reviews seeks to provide a comprehensive understanding of ashgard's health benefits and its potential for future applications in functional foods and herbal medicines. Introduction. The plant commonly referred to as Benincasa hispida or Benincasa cerifera is a member of Cucurbitaceae family. It is a widely grown vegetable crop, particularly in Asian nations, used for both biological and medicinal uses. The entire plant has therapeutic uses. The plant grows every year. This plant may sprawl on the bottom or crawl with the branch tendril that will climb over with the support or cover frame. Thick hairy stems have noticeable grooves and are aligned with the sharp bristles. The leaves have a greater rough surface and are spherical and kidney shaped. These have gorgeous golden yellow flowers. Various white colored seeds are inserted in the fruits 1 to 3 notches. The plant was utilized medicinally to treat the number of ailments including diabetes mellitus, respiratory conditions, heart condition, urinary disorders and gastrointestinal issues. Phytochemicals such as alkaloid flavonoids, tannins, glycosides, phenolic compound, amino acids, steroids, triterpenoids, and saponins are all found in plants. Because of their strong inhibitory properties, polyphenols can lessen the negative consequences of the conditions like infections, hepatic, cardiovascular, cancer, and neurological disorders. Tannin significantly reduces the activity of pancreatic lipase and intestinal fat absorption. The fruit's methanolic extract has been reported to include anti-ulcer, anti-inflammatory, antihistaminic, antidepressant, and bronchodilator, which are analgesic, diuretic, and antihistaminic drugs. Additionally, dry saccharides such as glucose, sucrose, maltose, etc. are also present. Taxonomy. Botanical name Benincasa hispida thumb cogn. Synonym ashgard, waxgard, winter melon, petha, kohla, white pumpkin, kshirfal. Biological source. It consists of dried fruit and seeds of Benincasa hispida thumb. Family Cucurbitaceae. Kingdom Plantae. Phylum Tracheophyta. Class Magnoliopsida. Order Cucurbitales. Genus Benincasa. Species Benincasa hispida. Cultivation. The major crop yield is produced in June and July, while the early harvest is produced in March and April using a variety of seeds varieties. Humidity, warm temperature, and well-drained soil are usually required for ashgard growth. Areas with an average temperature of 25 to 35 degrees Celsius and 150 to 250 centimeter of annual precipitation are suitable for its growth. The ideal temperature range for vegetative development and fruiting has been found to be between 32 and 40 degrees Celsius. The end of the underground is when tubers start to sprout. These sprouts vary in when they blossom and when they bear fruit. Morphological characteristics of fruit. shape oval to cylindrical, size can grow up to 60 cm in length and 30 cm in diameter. Skin smooth, waxy texture, typically pale green to white. Interior contains a white fleshy pulp with numerous seeds. Morphological characteristics of leaves: shape broad and heart shaped, size typically 10 to 20 cm long, margin serrated or slightly lobed, color bright green. Morphological characteristics of seeds: appearance flat oval shaped, color typically white or light brown, size small and numerous. Morphological characteristics of stems: growth habit climbing or trailing, length can extend several meters, texture green and may have a light fuzz or hairiness. Morphological characteristics of flower: color usually yellow with some variations, structure bell shaped or large flowers, arrangement can be solitary or in a small cluster. Chemical constituents: fixed oil and fatty acids constituent linoleic acid, oleic acid, palmitic acid. Pharmacological activity of linoleic acid: anti-inflammatory, cardiovascular health support. Oleic acid: antioxidant support, heart health. Palmitic and stearic acid: emollient, supportive in wound healing. Protein and amino acids constituents: globulins and albumins, various amino acids such as arginine, glutamic acid. Pharmacological activities: protein support tissue repair and immune function. Arginine has been studied for wound healing and cardiovascular health. Flavonoids constituent camphorol, quercetin, myricetin. Pharmacological activity of camphorol: anti-inflammatory, antioxidant, anti-cancer. Quercetin: antioxidant, support immune function, reduces allergic reactions. Myricetin: antioxidant, anti-inflammatory, may aid in neuroprotection. Sterols constituents: beta cytosterols and stigma sterols. Pharmacological activities: beta cytosterols, anti-inflammatory, support cholesterol management. Stigmasterol: anti-inflammatory, may support joint health. Triterpenes constituent: cucurbitacins, especially cucurbitacin B and cucurbitacin D. Pharmacological activity of cucurbitacin B: anti-cancer, anti-inflammatory. Cucurbitacin D: antioxidant, anti-inflammatory properties. Phenolic compounds constituent: gallic acid, ellagic acid. Pharmacological activities of gallic acid: antioxidant, antimicrobial, supports liver health. Ellagic acid: antioxidant, anti-cancer, may protect cell from oxidative damage. Saponin constituent: diosgenin. Pharmacological activity: anti-inflammatory, support immune response, cholesterol lowering effect. Minerals constituent: magnesium, calcium, zinc, iron. Pharmacological activities: zinc support immune health, wound healing and antioxidant enzyme activity. Magnesium and calcium aid in bone health and muscle function. Carbohydrates constituent: dietary fiber, simple sugar. Pharmacological activities of dietary fiber: supports digestive health, regulates blood sugar level. Vitamins constituent: vitamin C and vitamin E. Example: vitamin C. Pharmacological activities: vitamin C: antioxidant, immune support, promotes skin health. Vitamin E: antioxidant, supports skin and heart health. Here are some chemical structure of Benincasa hispida constituent. Microscopic characteristics: fruit. The TS of fruit has a single layer of epidermis composed of rectangular parenchyma cell with a waxy ash covering the outer surface. This is followed by the several layers of hypodermis made up of tangentially elongated, thin-walled parenchymatous cell with starch grain embedded in them. In a mature stage of fruit, 4 to 6 layers of stone cells run continuously to next to the hypodermis, but in a tender stage, they are found in patches. Mesocarp, parenchyma cell of varying size, come next. The size of the cells rises from the periphery to the inside of the mesocarp. The mesocarp is extremely watery and the cells have oval shaped and thin walls. Here are the microscopic images of the microscopic characteristics of the fruit. Microscopic characteristics of seeds. TS of the seed displays a slimy outer layer of epidermis followed by the thin walled outer scleroid of delicate seed. In a center of cotyledon portion which has loosely arranged parenchyma cell in a tender seeds and compactly arranged palisade parenchyma in a mature seeds are the stone cells, peri- perisperm, endosperm and collapsed cell layer. These cell contain starch grain. Here the images of the microscopic characteristics of seed. Extraction process. First, residue and fresh juice supernatant. The fruit's cuticle was removed in order to extract the juice using an electrical juicer. A T8 electric centrifuge was then used to centrifuge this mixture juice for 10 minutes at 5000 rpm. Following the aspiration of the supernatant, both the residue and the supernatant were gathered and kept apart for use in further experiment. Alcoholic extract. To make an alcoholic extract, 1000 milliliters of fresh juice and 5000 milliliters of ethanol or methanol were combined and let to sit at a room temperature for 36 hours. After 2 hours, the intermittent stirring, the slurry was left overnight. Following the filtration of the mixture, the filtrate was allowed to dry for 24 hours at a room temperature, such as 20 to 25 degrees Celsius. Later, a lipophilizer was used to thoroughly dry the extract. The extract's yield was 1.27 gram per 100 ml of fresh juice, and the end product was an off-white powder. Petroleum ether extract. In order to prepare this extract, 1000 milliliters of fresh Benincasa hispida juice were extracted using 90% of petroleum ether in a Soxhlet device for up to 4 cycles. The filtrate was dried with a lipophilizer after passing through Wattsman filter paper number 40. 1.73 gram of fresh juice per 100 ml of was the yield. Physical activity, anti-inflammatory activity. The research shows that when Benincasa hispida fruit is extracted using methanol and petroleum ether, it has anti-inflammatory qualities. Both extract efficiently and dose-dependently decrease the rat's model paw edema caused by the carrageenan, granuloma caused by cotton pellets, and paw edema caused by the histamine at a dose of 300 mg per kg body weight. Rats that were exposed to inflammation caused by egg albumin were able to tolerate the anti-inflammatory properties of fruit peel methanolic extract. Antioxidant activity. The methanolic extract of Benincasa hispida seed showed a significant dose-dependent free radical scavenging effectiveness in comparison to the ascorbic acid. The extract shows the maximum amount of radical scavenging activity calculated to be 79.8% at a concentration of 300 mg per ml. At a dose of 200 mg per ml, the extract demonstrated 63.7% H2O2 means hydrogen peroxide scavenging activity. Methanolic and aqueous extract of Benincasa hispida showed significant neuroprotective potential by reducing the oxidative stress brought on by the injection of D-galactose. Anti-asthmatic effect. From the methanolic extract of Benincasa hispida fruit, two triterpenes, aleunosenol and multifluorenol, showed a stabilizing effect on mast cell and were found to have the capacity to limit the release of histamines caused by antigen antibody response. Even at a relatively low oral doses of 50 mg per kg, methanol extract of Benincasa hispida demonstrated good protection against histamine-induced bronchospasm in guinea pig. But even at a high dosage of 400 mg per kg, MEBH did not significantly prevent acetylcholine-induced bronchospasm. The findings imply that the protective effect against histamine aerosol-induced bronchospasm can be mediated by the histaminic action antagonism of H1 receptor. Anti-ulcer effect. Methanol and petroleum ether extract of Benincasa hispida were also studied for their anti-ulcer properties in rats. Oral doses of 300 mg per kg between of petrol petroleum ether and methanol extract and 20 mg per kg body weight of omeprazole, the reference standard, were administered. The two extracts significantly decreased the ulcer index in all models such as ethanol-induced stomach mucosal damage and other gastric ulcer model, and the outcomes were similar to those of the group that received omeprazole treatment. Moreover, a noteworthy decrease in vascular permeability was noted. Antihypertensive effect. Due to its ACE inhibitory activities, the herb may have pharmacological foundations for its long standing traditional Chinese medicine medical applications in treating high blood pressure. Fruit juice 0.4 to 1.6 ml per kg intravenous released nitric oxide from the cultured swine aortic endothelial cell shows concentration dependent relaxation of isolated rat aortic ring and decreased blood pressure in dose dependent. Diuretic activity. Adult male guinea pig were used to assess the diuretic effect of an ethanolic extract to 20 to 200 mg per kg from the fruit rind of Benincasa hispida. The extract caused a notable rise in urine volume, while potassium excretion substantially decreased, whereas the sodium and chloride excretion dramatically increased. Anti-diabetic effect. In rats with alloxan-induced diabetes, a methanolic stem extract 50, 100 and 200 mg per kg per once reduced blood glucose levels in dose-dependent manner. After 14 days of therapy, the chloroform fruit extract 250 and 500 mg per kg per once improved the lipid metabolism abnormalities in alloxan-induced diabetic albino rat in a dose-dependent manner. According to the study, methanol, ethanol, and aqueous peel extract has had considerable alpha amylase inhibitory activities. In a dose-dependent manner, the diabetic mice's blood glucose level were reduced by the ethanol and ethyl ethanoate leaf extract. Anticonvulsant activity. The fruit methanol extract 0.21 gram per kg per once demonstrated dose-dependent anticonvulsant efficacy in a maximum electroshock model strychnine and picrotoxin and pentylene tetraazole. In contrast, the methanolic extract of fruit peels and had an anticonvulsant effect on mice model of a convulsion caused by the pentylene tetraazole that was dose dependent 0.25 to 1.5 gram per kg. Swiss albino mice exhibited dose-dependent anticonvulsant efficacy when given ethanolic seed extract 250 and 500 mg per kg per once. Effect on Alzheimer's diseases. The fruit extract administered at a dose of 400 mg per kg per once protected rats with Alzheimer's disease induced by the colchicine, possibly because it contained both vitamin E and beta carotene, which protect rat neurons from oxidative stress. In contrast, in the colchicine induced Alzheimer's rats model, The aqueous fruit pulp extract 100 to 450 mg per kg dose dependently raise SOD superoxide dismutase catalase and while decreasing LPO levels. Skin anti-aging. A cream made from the dried fruit pulp extract, petroleum ether, chloroform, ethyl acetate, and methanol of Benincasa hispida shown a notable anti-aging effect on stratum corneum of human skin and densyl chloride fluorescence model. Here are some images of marketed preparations of Benincasa hispida, such as juice and ashgard juice by the health as a health supplement. Conclusion. Due to the fact that allopathic medicines have more negative effect and that this herbal remedy has many actions to treat various illness, there is a growing demand for herbal medicine plant worldwide. All of the extract of Benincasa hispida contains tannins, saponins, terpenoids, phenolic compounds, and alkaloid according to the phytochemical analysis. The above mentioned phytochemicals of Benincasa hispida extracts give the therapeutic qualities. So we come to the conclusion that Benincasa hispida exhibit anti-anthelmintic, anticonvulsant, antioxidant, antihistaminic, anti-cancer, antibacterial, anti-inflammatory, and analgesic, diuretic, anti-asthmatic, central nervous system action, anti-ulcer, and anti-diabetic properties in an animal models based on these investigations. Because of its pharmacological properties, Benincasa hispida is a well-known plant in research literature. Here are some references. Thank you.